everyone, it's your teacher here again, Mr. Jennings, for yet another general biology video. Today, we are going to talk about the extensions of Mendel's. What happens when Mendel is wrong? So, last video we talked about Mendel has four basic things, discoveries, laws, whatever. One is that you inherit traits from your parents, and those are called genes. My kids are screaming again. So, Mendel inherits. You inherit genes from your parents. He didn't call them genes, but he said you inherit traits from your parents. Two, the law of dominance, that whenever a dominant trait is present, it is going to override any other trait. Those are called, recessive traits, the traits that are overridden. So some traits are dominant to others, and the others are recessive. Law of dominance. Third rule is law of independent assortment, that you only get one allele from each parent. And then the third law, the fourth thing he discovered, though, is the law of independent assortment, that traits are independently assorted, and the inheritance of one trait does not influence the inheritance of another trait. For instance, if you have a pea plant, your seed shape does not mean your seed color will be different. It doesn't affect how your seed color is. So in the example they used, you could be a round yellow pea plant and have offspring that are round in green, wrinkled in yellow, and green and wrinkled, as long as you cross with another round yellow dihybrid. Alright, so today we're going to talk about what happens when Mendel is wrong. So, a little bit of background here is that Mendel, and once we do that, we're going to go ahead and zoom down here. Mendel here didn't understand modern genetics. It's part of the thing, because modern genetics wasn't invented. He was a monk in Austria-Hungary. That country does not even exist anymore. So, that's how long. Alright, so we're talking about Mendel here. So these are some important exceptions to Mendel's important principles. Some alleles are neither dominant nor recessive, and some are controlled by multiple alleles or even many genes. So just keep that in concept here. Mendel didn't understand a lot of this stuff because, one, he got really lucky. He picked a plant that picked very simple traits. He got lucky. A lot of traits actually do not follow the Mendelian pattern at all. Turns out most traits are what we call non-Mendelian, but Mendel still forms a good basis. So we see that a lot. So first up is incomplete dominance. This happens whenever one of those is not completely dominant over the other. And this actually results in the blending of traits. So one of the things Mendel sought out to disprove was the idea that traits blended. Well, this is one of those traits that do blend, but they don't stay blended. They're blended because the two traits, in this case the red snapdragon and the white snapdragon, when they get crossbreed, you produce a pink snapdragon. Hold on. Sorry for the interruption. You didn't see that pause anyway. But it's not a true blend, because if I cross two pink flowers, across these two pink flowers, I would get a red and white flower. So it's not a true blending. It's just that when put together, they express a combination of the traits. So back to what I was saying. For example, homozygous red flowers cross with white flowers, and you get all pink flowers. And you get three different genotypes along with three different phenotypes. And we don't use lowercase letters when we do incomplete dominance because the main reason, there's no trait is neither really dominant, so we don't use recessive letters. Alright, codominance. This is when both alleles contribute to a phenotype, and we call that codominance. Now, colors are not blended. Instead, they appear separately. For example, in some varieties of chickens, like we see here, the black allele is codominant with the white feather allele. The chickens have feathers that are both speckled and black and white. Speckled, black, and white with no actual blending. It may look kind of gray, but if you actually look at individual feathers, you're going to see black and white individually. There is no blending here. Another example is human blood type. We usually use, for codominant situations, we use the common letter, in this case I, and we use in the exponent position, we would put the actual allele. So for instance, IA is blood type A, IB is blood type B. They're codominants. So when you have A and B together, you have both A and B blood types. You don't have a mixture of the two. You have two distinct blood types at the same time. Now, some traits are what we call multiple alleles. This is not the same as incomplete and codominant. This is multiple alleles for one trait. This means there's more than 2 possible alleles for a population. However, only 2 alleles are inherited. So, for instance, here in this chinchilla or rabbit fur is controlled by 4 different alleles. So here, they all use common lettering, and it does show one is dominant to the other, but the exact combination of them result in different traits. Now this is contrasted with polygenic. So there's multiple alleles. Also another example of multiple alleles is blood type, human blood type. We have A, B, and O. So we have 3 alleles. More awkward. Okay, so the deal is in human blood type we have A, B, and O. So whenever you change the blood typing of humans, or look at the blood typing of humans, you're going to see that. And this is what we see in the rabbits here. There's 4 different alleles. They all influence the trait. Now contrast that with polygenic traits. Polygenic traits is when multiple genes control two or more genes control a trait. For example, skin color and eye color in humans are controlled by a variety of genes. And they control those traits. Different combinations of alleles yield a very enormous range of variation in skin color and eye color. Here's the eye color, for example. There's no one true blue eye color, and there's no one true brown eye color. It's a spectrum. As the traits are inherited, as the multiple genes are inherited, each different gene contributes a different pigmentation. And a lot of traits are actually polygenic. Things like height, weight, eye color, hair color, skin color, they're all polygenic. So anything there's no distinct categories. For instance, in hair color, there's no one red hair, there's no one brown hair, there's no one blonde hair. There's variations of them in between. Those are influenced by all the different traits you inherit. Some characteristics are also determined not by just genes, they're also determined by the interaction between genes and the environment. So height and weight also are influenced by your environment. Example, PKU. PKU is a genetic disorder that can cause mental retardation and other health issues. And wealthier countries have the ability to test for high levels of PKU during pregnancy. The mothers can be put on a special diet to lower the PKU level. However, in poorer countries unable to perform the tests, there are higher levels of this rate. So what we have to do with PKU is basically they have the inability to process certain sugars, so they have to go on very much an aggressive keto diet. Okay. So, sorry, my sons are here. Want to say hi to? Doesn't want to say hi. Okay. Nothing wrong with that. So here's the deal. As you have to go through a big keto diet. There was a hard pause there. As you go through a big keto diet, they basically control the environmental factors control the severity of the genetic disorder. We're talking about genetic disorders. Now, if genes are located on the same chromosome, because remember, genes are found on chromosomes, are they inherited together? And yes, Thomas Hunt Morgan was the first one to realize this when he was studying fruit flies. The fruit fly, specifically Drosophila melanogaster. Now, this fruit fly here is realized that many of the genes appear to be linked. Basically, that means they're inherited together. This led to 2 distinct discoveries. 1, each chromosome is a group of linked genes. And 2, it is the chromosome that independently assorts, not the individual genes. So that was actually the major refutation. That was the major refutation of Mendel. Mendel thought genes inherited independently and independently assorted, but it turns out it's the chromosomes. You inherit 1 chromosome from your mother, one chromosome or DNA. Now, you inherit a chromosome from your mother and a chromosome from your father, and each of those chromosomes you inherit contain a package of genes. And those genes are what's independent. The chromosomes are independently assorted, not the genes themselves. Now, if 2 genes found on the same chromosome, do they mean they're linked forever? Nope. Because of prophase 1 crossing over, during meiosis, the genes can separate, and they usually were found on the same chromosome or not. Crossover events. Hold on, just a second. A little softer. Alright, so crossover events exchange separated genes and produce new combinations. And this is where we get a lot of genetic diversity. So this is what we call genetic recombination. This brings up the concept of a gene map. One of the students of Thomas H. Morgan, Alfred Sturvant, he wanted to find the rate at which genes crossing over separated linked genes. He hypothesized that the further apart the two genes were, the more likely they were to be separated by crossing over. And this rate then would produce a map of distances between genes. And this is where we get what's called a gene map. Now, gene maps are not 100% accurate, but they are pretty dang close. And this method is used to help construct maps, including maps of the human genome, so we know where genes are found on our DNA. An example of this is the Drosophila gene map. These are relative based on how frequent crossing over occurs. That's a fruit fly, buddy.
I don't know.
They're tiny little flies that fly around. They're actually much smaller than that.
Hey, Dad, by the way, about the common because and then the thing came off because it was still sticking. It's sticky fit.
Let me finish up, okay, buddy? So, let's wrap things up here. Alright, so when it comes to Mendel didn't understand modern genetics because the concepts weren't there yet. What he did understand and found the foundations for was how genes are inherited, and his work was revolutionary for setting up how genes are done. Hey. Alright. No, I'm trying to finish up here, buddy, okay? Well, we can finish in a second. Now, sorry for this. Are you going to even say hi to the camera, Jude?
Why?
Because the students may want to know who you are. His name's Jude. Alright, so to wrap things up, Mendel didn't understand modern genetics. However, modern advancements and understanding how traits are inherited shows that the world's actually much more complicated than what Mendel originally proposed. And while we do know that Mendel's ideas are flawed, doesn't mean they're wrong. Just they're really simplistic, and there are simplistic traits out there. No, it's not broken. You just squeeze it. And that's called a hinge, buddy. It's not broken.
It's almost. I can see it coming. I can see it up.
That's normal. Sorry, we're looking at a toy. Alright, well, let me finish talking, okay? Alright, so when it comes to these concepts, even though most traits are not Mendelian in nature, most are going to be, something like that. And the concepts of Mendelian genetics are really important for forming the basis of understanding the more complexities of genetics. Alright, next time when we talk, we'll be doing, I'll probably do that Punnett Square video if you haven't already. And we'll then talk about pedigrees, which is actually where the state standards are going towards. But we had to explain all this basic information to understand the pedigrees. Yeah, thank you for watching. Hope you're staying safe out there. Be careful. I'm Mr. Jennings. Have a great day.